Hello, welcome, and thank you for listening to my lecture about cells, which is chapter 4 in your textbook. Today I'm going to briefly revisit cell theory. We're going to talk about different types of cells and then cell parts and their functions. There is one last part starting in section 9 of this chapter about cell transport, diffusion, and osmosis, and I'm going to save that for a short follow-up lecture. So to briefly recap cell theory, it has 3 parts. The first part is just that all organisms are composed of at least one cell. The second part is that cells are the smallest living units. And then finally, the last part is that cells come only from other cells. Okay. This is just a figure from your book showing the different sizes of different cell organelles and cells. You may look at that in your book. It's figure 4.1. Okay, so cells are not all the same size, but most of them are small because larger cells are not as efficient as smaller cells. And part of the reason for that is that in large cells, there's a greater distance between the nucleus or the command center and the edges or the cell membrane. So organisms that have lots of small cells are more efficient at functioning than those that have fewer larger cells. And one thing that does make smaller cells more efficient is that they have a greater ratio of surface area to volume. And the surface area or the cell membrane is the only point of contact that the interior of the cell has with the environment. It's the place where anything that the cell needs from the environment is brought into the cell, or anything that the cell needs to get rid of can be gotten rid of. And so as cell size increases, the volume increases more rapidly than the surface area. And so that's another thing that makes larger cells less efficient, is that they have a lower surface area to volume ratio. Okay, so we have visited this briefly earlier in the semester. The 2 cell types are prokaryotic cells and eukaryotic cells. And the major difference between the 2 cell types is that prokaryotes do not have a true nucleus. They have genetic material, but it's not enclosed in a membrane. And then prokaryotes also do not have organelles, and they don't have an extensive or well-developed system of internal membranes. The 2 domains that have prokaryotic cells are bacteria and archaea. And then the domain eukarya has all of the kingdoms that have eukaryotic cells. So plant, animal, and protists are in the domain eukarya. So eukaryotic cells have a nucleus that is surrounded by a nuclear envelope or a nuclear membrane. And then the eukaryotes also have a well-developed system of internal membranes, including organelles that are surrounded by membranes. Organelles are cell parts. And then all organisms except for bacteria and archaea are eukaryotes. So starting with prokaryotes, these are the simplest organisms. They have a plasma membrane surrounding them, and sometimes they have external layers to the plasma membrane, such as cell walls and capsules. In bacteria, the cell walls are made of carbohydrates and proteins and give a rigid protective structure, and then capsules are made usually of carbohydrates, and they can add a layer of protection outside of the cell wall. So the inside of prokaryotic cells have a very simple structure. There's cytoplasm with little internal support, and then they have ribosomes that are just free floating in the cytoplasm. Ribosomes are the sites where proteins are made. And then there's a nucleoid region, which is where the DNA is found. And the DNA, or like we said before, the nucleoid region is not surrounded by a membrane, so it's not a true nucleus. So other structures that are found in prokaryotes are flagellum, which the plural of flagellum is flagella, which is a thread-like structure made up of protein fibers that projects from the cell surface and usually is used for movement and feeding. And a single cell can have one or many flagella. And then the other structure that I wanted to talk about is called a pilus, which pili is the plural of pilus. And that's just more or less a short flagellum. Usually they're used for attaching to surfaces or exchanging genetic information with other cells. So this is just a picture. This is figure 4.5 from your book, and it's just showing the general structure of a prokaryotic cell. You can see these are the pili. The little round circles are meant to represent ribosomes. This area where the DNA is found is the nucleoid region. This is a picture of a flagellum. This white area on the very exterior is the capsule. The purple area surrounding it is the cell wall, and then inside of the cell wall you can see the plasma membrane, and then everything that's inside of here is just in the cytoplasm. Okay, so moving on to eukaryotic cells. Eukaryotic cells are larger and more complex than prokaryotic cells. They're surrounded also by a plasma membrane, and they have an internal membrane system that form compartments, which we can think of as organelles or cell parts. And each of the organelles has a specific function in the cell. And then within, this is a difference between eukaryotes and prokaryotes also, the cytoplasm has a network of protein fibers, which we call a cytoskeleton. And the cytoskeleton helps to provide some structure for the cell, and it also helps in moving things and in cell division. So many cell organelles can be seen easily through a microscope. The nucleus, which eukaryotes have a true nucleus, is a membrane-bound compartment that contains the DNA. The endomembrane system is what generates the internal membranes found in the cell, which we talked about earlier a little bit. So there are different types of eukaryotic cells. The cells of plants and fungi and some protists have a cell wall surrounding the plasma membrane. And all plants and some protists have chloroplasts, which we'll talk about in a little bit. Plants have a central vacuole, but animal cells, fungus cells, and protists do not have a central vacuole. And then only animal cells contain centrioles. So this figure 4.6 from your book shows the basic structure of an animal cell, and the first thing that should jump out at you is that it's a lot more complex than what we looked at when we were looking at the picture of the prokaryotic cell. So we're going to be talking about some of these individual cell parts in a little bit, but this is a really good figure for you to look at to become familiar with what the different cell structures look like. This is a figure 4.7 from your book, that shows the structure of a plant cell. And so between this figure and figure 4.6, where you see the structure of an animal cell, you can see some of the differences between the two cell types. In the picture of the animal cell, you can see there's no cell wall, there's no central vacuole, but then there are centrioles right here. And then in the plant cell, you can see this big, giant central vacuole, and then you can see the cell wall surrounding the plasma membrane. Okay, so the way that we think of the structure of the plasma membrane is the fluid mosaic model, which I will let you read about in your book in section 4.4. And then also we talked about this when we were talking about macromolecules, but the main thing making up the plasma membrane is phospholipids, and we call the general structure of the plasma membrane a phospholipid bilayer, and it has proteins embedded in it. And so this is also section 4.4, but just a brief review. Phospholipids have a polar phosphate head and nonpolar fatty acid tails too. And so the polar region is water soluble or hydrophilic, and the nonpolar region is non-water soluble or hydrophobic. And so because of this structure of the phospholipids, whenever there's a collection of phospholipids or whenever it's placed in water, they self-assemble into a phospholipid bilayer. So the interior part of the bilayer is completely hydrophobic or nonpolar, which makes it to where hydrophilic molecules cannot cross easily or freely across the cell membrane or the plasma membrane. And then another thing about the structure of the plasma membrane is that it has cholesterol molecules in it, which help to keep the membrane fluid, not rigid or crunchy. Okay, so another component of the plasma membrane are membrane proteins. Some of them cross all the way through the plasma membrane, and they're used as channels. They're called transmembrane proteins, right here, transmembrane proteins, and they form channels or tunnels that allow molecules to cross through. And then there's another type of proteins associated with cell membranes, and they're called cell surface proteins, and usually they're attached to the outside of the plasma membrane, and they can act as markers to identify cells. So this is just a picture of the plasma membrane with the proteins, and these are this is the phospholipid bilayer, 2 layers of phospholipids, protein channel, and these are cholesterol molecules. And then this is a cell surface protein that acts as a marker. So the next cell part is the nucleus, and we're doing eukaryotic cells right now. We know that because they have membrane-bound nuclei. So the nucleus contains the DNA. It's thought of as the control center of the cell, and it's surrounded by a double membrane, which is called the nuclear envelope, sometimes the nuclear membrane. And there are groups of proteins in the nuclear envelope that form openings or pores called nuclear pores, and those allow proteins and RNA to move in and out of the nucleus. So the DNA of eukaryotes is in linear segments called chromosomes, and the chromosomes also contain proteins. Proteins allow the DNA to be wound up tightly and condense into chromosomes during cell division. When the cell is not dividing, the chromosomes unwind and unravel, and they're kind of just thread-like strands in the cytoplasm or in the nucleus called chromatin. So protein synthesis happens when DNA is in the chromatin form. Okay, so ribosomes are where cells build proteins. Ribosomes are made of ribosomal RNA, also known as rRNA, and proteins. And the ribosomes are made in the nucleolus, which is a region inside of the nucleus. Okay, so this is just a drawing of the nucleus, and you can see the nucleus with the nuclear envelope, and then the nucleolus, which is where ribosomes are made. And then the nucleoplasm, which is the nucleus's version of cytoplasm, which has the chromatin, or DNA. Okay, and so the last thing I wanted to point out in the nucleus are the nuclear pores right here, which are made from proteins. I'm going to stop right here because I'm going to run out of time on this lecture, but I will do a part 2 for chapter 4 and also a part 3. So you can look for that later today. Thank you.